Okay, so lecture 28. So Matt, you have a presentation for us? You'll need the document can?
Yep.
Okay, so.
So I did a crossword about metabolism and energy processes, the FGPTO process. And so kind of just to help you remember that and remember what goes into each process. I was
Switch back.
switched.
Put the document cam button on where Reese is pointing. For a reason, switch back.
So just 10 questions, crossword, pretty
Okay.
self-explanatory. I have an answer key too, but I don't want to give out all the answers and have everyone cheat on it.
Okay, so you're gonna, so potentially, does anybody want this for over the weekend to study?
Yeah, I'd take one.
So in the break, we can make a copy of what we got here and then is the answer key on the back or?
I have it on a separate document.
Okay, separate sheet, so we won't give that out. We'll
Oh,
save that for Monday or something like that. So
yeah.
I would imagine if you get it done, it probably works, right? So. Okay. Any questions for our speaker? So during the break, I'll copy that and we'll take it from there. Or somebody has a copy of it.
I'm gonna leave it up here. isn't it?
Yes, please. Yay. And there was much rejoicing. They ate Robin's minstrels, right? You guys don't know that movie, but anyway.
I'd love to hear your motivation for that sometime. I'd love to hear where the motivation from that project came from, the inspiration.
Inspiration?
Yeah.
Learning.
Oh, okay.
Help other people.
Okay, so he's a generous giving kind of guy, right? So assignments, I think I got the dates right there. So 13 and 14 are posted. They are extra credit. You turn them in. You've got one more assignment in the bank. So you can do those. I guess that's chopped off at the bottom. So written assignments for the exam will be, I think we start somewhere in 9 and then we go through 14. So I think I've taken all the assignments related to, I think we start. Nope, that's the other class. We start there. So lectures, 19, 20, 21. Then we skip over 22, which was our last exam, 23, 24, 25, 26, 27, and 28. So do you guys want any sort of review time?
Yeah.
Okay. What would you like? When is it, did we agree to 4? I can't remember.
Yeah.
4 on Tuesdays, give or take in here.
Yeah.
I assume it'll be open. I guess I should double check. That was interesting. Wonder what they're going in there to do. Okay, so 4 o'clock here. When do you guys want to review? Night before? Monday? I think I've got a 1:30-ish exam that should be done by 4. There's some students that take 2-3 hours to take it. Just because that's what they like to do. So anytime after 4 on Monday?
I think there are 2. It was 200, stuff was 4 to 6-ish. So
Okay, so after that? 6, 7? How many people would like 6? Or should we be safe and say 7? You can go get dinner and come back? Okay, so review time will be 7 o'clock. Monday, what's the date? The 12th? May 12th at 7 PM in Marshall Hall. Do I need to put that up on Teams or everybody can do it on their own? Okay. Okay, so any questions or concerns? Yeah. Is it visible or not? Did I make it visible? Let's double check. I thought I did. Right space. Login. Recorded lectures on YouTube. So sometimes you have to go down here to this load more. Because it, for whatever reason, seems to limit the amount. So 27 is up there. I don't know as I got to it till late on Tuesday. I had some problems downloading it, so it's there now. So, exam for the final, I guess you guys can't read that, can you? That can be very embarrassing. So we talked about hormones in cows. We had that list last time. I printed that out. I gave it in the front of this handout, so the list is there. We talked about matching for that. Or no, you guys, I give you the definition, you give me the hormone.
yeah,
Is that the way we're doing it? Okay, so those are the ones I can choose from. So hormones in cows. Let's talk about a little bit about what hormones do. So the first thing we'll talk about is lactation. So lactation is unique to mammals. So mammals, the name comes from the mammary gland. So all mammals have a mammary gland, even a platypus, which is kind of odd since they lay eggs, but they still nurse their animals after they come out of the egg. So two purposes. Did I have that in there somewhere? It's in our last handout, right? For the end of our there. So two purposes for we're thinking about our cows. Provide a source of passive immunity to the newborns. So that lactation, that first milk, that colostrum is going to provide that opportunity. We don't necessarily need it in humans because we get a lot more in utero transfer of that passive immunity. There's less layers to the placenta. So we have 3 layers contributed from mom in the uterus, 3 layers contributed from the embryo, and some of those start to erode away, and we're down to 1 or 2 layers, I think, in a human. Those layers of skin or whatever you want to call them, layers of tissue. And then for our cows, it's almost perfect nutrition for the young. If we looked at, we were discussing the cost of raising a heifer in calves and heifers today. if you look at the efficiency of gain for that calf on milk, it's like 4 times the efficiency that we get later in a heifer. So pound of gain for every pound of dry matter, we're looking at 60% efficiency with milk-fed animals, and we drop to 10% as we get into older animals. Those proteins, mixes, and stuff associated with our plant materials are not nearly as efficient, not nearly as perfect as our milk proteins. So we're going to produce milk up in the secretory tissue in the top half of the udder. You're going to have an organization. This is a microscopic approach here where alveoli are lined with secretory cells. Milk is secreted in the middle, and then we push that milk down to the lower part of the udder into the gland cistern, into the teat cistern, and there's a certain amount of mixing that's going to occur along the way. So you have a basement membrane that lines the outside of the alveolus. The secretory tissue is on the inside. Technically, that secretory tissue is on the outside of the body. You could, in theory, if you were small enough, go in the teat end and end up standing in that alveolus without drawing any blood. Around the alveolus, you've got the muscle. So you're seeing the muscle here that will contract that alveolus. And then you got the blood supply, the capillary bed on the outside. So the blood supply, everything diffuses across that membrane into those secretory cells. And that's where the manufacture of milk protein, lactose, and milk fat occurs. We throw all that stuff into the lumen of the alveolus, and then water follows passively until we get our final milk volume. So using text, alveoli aligned with alveolar epithelial cells. Those secretory cells take raw materials from the blood on the other side of the basement membrane and use that to make milk. During milk letdown or the milk ejection reflex, those alveoli are squeezed and the milk goes down the duct system. So why do we have to do that? If I take a straw, playing with my soda, right? Straw, put my finger on the top, lift up, what happens? Soda stays in the straw. If I take my finger off, falls out. We don't have a hole at the top of the alveoli. It will not come out with gravity. We have to squeeze it out. So that's why we need that milk ejection reflex, and then the milk moves from a high to a low, high up in the top to the lower pressure down below. So milk let down review. Oxytocin is released from the where? On your hormone list. Where's oxytocin released from? Posterior pituitary. So we have that stimulation of the teats. So calf nudging the udder is one way to stimulate that response. Another is the act of putting animals in the parlor. Some of them will start to say, hey, I know what's going here. They will start to release oxytocin, start dripping by the time they get in there. But washing and cleaning the udders, particularly with those terry towels, seems to promote a good milk letdown response. The milk letdown response or reflex. Then we've got the oxytocin will be blocked by adrenaline or epinephrine. So the stress hormones, where does that stress hormone, adrenaline, where is that released? The adrenal medulla, the center of the medulla, or center of the adrenal gland. So it's going to block oxytocin two ways. It seems to compete for the binding sites on the secretory tissue. Adrenaline and epinephrine will also reduce the flow to the skin as part of the fight or flight response. And if we think about the udder is part of the skin embryonic tissue. So if we slow the flow of blood to the udder, we will also slow the flow of oxytocin to the udder. You'll also, over time, you'll also reduce the nutrients for milk synthesis and produce less milk if you stay in that sympathetic state. Cows are always afraid they're not going to produce as much milk. So we've got two parts of this milk ejection reflex. First, we have the nerve impulse, goes up to the brain, goes up to where in the brain? Can anybody guess? Hypothalamus, which is going to send the axons down into the posterior pituitary and then release oxytocin from those axons into that capillary bed. And then oxytocin is going to flow through the blood. It's going to go to the heart. Then it's going to go to the lungs and back to the heart, and then get pushed out through the body and hopefully find its target tissue in the mammary gland. Once it's there, it attaches to those myoepithelial cells that band around the alveolus and hopefully squeeze down that alveolus to push the milk down the duct system where it can be harvested by a calf or a milking machine. So nerve impulse, how long does that take? Right? Signal goes directly to the brain in a fraction of a second. We have to wait to attach those machines. How long after stimulation? So minute and a half is a good average. It used to be a minute. Now we know it takes 2 minutes. Why does it take 2 minutes? Minute and a half, minute to 2 minutes. We want to attach that machine after stimulation. Why is that? Bloodstream. It takes 2 minutes for that to go into the capillary bed, down the jugular, vena cava, heart, lungs, heart, and then spread out aorta and goes all the way around the body. So you've got oxytocin in the blood all over the place, but it's only got those important targets in the udder. So it's the blood side that takes the time. So, and again, the heart to lungs and back is missing in this diagram. So you got the adrenal gland that's going to get there about the same time. If this is a frightening experience, if milking is painful, you're not going to get as strong an oxytocin squeeze. It's going to block things moving through. Any questions about the milk ejection reflex? So we go from a nervous response to a hormonal response. And the hormones are what takes the time. We want to catch that milk as it gets squeezed out. We don't want them put on the machines too early, or we'll be dealing with no flow of milk, and that's going to compromise and potentially damage the DANS. We don't want to wait till late, because if we get a backup in the system, we won't probably milk out as well as we should. So as that udder squeezes at the top, we want to attach the machine to catch that flow as it comes down. The benefits of timing that right, good stimulation, timing right, we should reduce our overall milking time. So we'll milk the herd faster, we hope, if we time everything correctly. If we don't do good stimulation, we'll get what are called bimodals. Has anybody ever heard of that before? So if you look at the flow rate per minute or pounds per minute, you're going to get a graph that looks like this. So we don't get a strong stimulation. We empty out the gland cistern, the teat cistern, and then we get a low flow until the machine takes care of the stimulation. And that means 2 minutes later, we get that big flow coming down. So we've extended milking 2 minutes, and 2 minutes on every cow. However many turns of the parlor you do per hour. If you're doing 8 turns or 4 turns an hour, that's going to add 8 minutes just because you're not doing your job right. So that can slow things down considerably. If we're thinking about milk production and milk synthesis, milk synthesis is under hormonal control. I'm sure we don't know all the ins and outs of how that happens. We have not been able to mimic by giving them the same hormones that we know about in the same concentrations, the galactopoetic, the ability to start milk production, that a pregnancy will. Pregnancy is the best way to set up and promote milk production. So mammary gland development is under hormonal control. Estrogen stimulates duct development in the mammary gland. Progesterone stimulates growth of the alveoli. So as that animal goes through cycles, you're going to have periods of alveolar growth and duct growth as that mammary gland matures. And then when we get pregnant, you're going to have a huge expanse, hypertrophy of those different features, especially as we get into the last 2 months of gestation. Others that we know are involved: FSH, LH, somatotropin, placental lactogen, and thyroid hormones all contribute positively to mammary gland development. Milk secretion. It gets started by somatotropin, adrenal corticoids, getting the glucose up so we can produce that lactose, and then a hormone called prolactin. It's not on your list, but just by its name, prolactation. There, milk secretion, once we've got it up and running, there are a number of hormones that are going to support that. We start out with high levels of somatotropin, oxytocin, prolactin, adrenal hormones, thyroid hormones. As our days in milk increase, we start to lose them in concentration. And then our milk secretion tapers off in parallel to that. The shape of the lactation curve is pretty much follows or mirrors the somatotropin curve, a daily average for a cow. Milk secretion is a positive feedback system. The more we withdraw from the udder, the more that will be secreted. So if we're not emptying out the udder, the whole process starts to slow down as we build back pressure. If you're going to dry an animal off, you need to build back pressure to shut down secretion completely. But this is meant to, I think, mimic calf. The more the calf needs, the more the calf wants, the more is secreted. And then we have that gradual tapering off on the other side, that animal starting to switch over to solid food. We might have the consumption might not be as high. And then as that back pressure continues, it will slow secretion until it stops. We think about the total milk secreted. We have the number of secretory cells in the udder, and we multiply that by secretion per cell. So the upside of the lactation curve going up is that change is a lot about getting more secretion per cell. As we go down the curve, we're losing less production, we're getting less production per cell. We're also losing cells as we go through the lactation. So numbers are going down, secretion per cell is going down. That's going to give us that negative slope on that lactation curve after peak. So we've got secretion per cell, certainly for the first part. At the beginning of lactation, that's the maximum number of cells we're going to have. And they don't get re-established until we get to the other side. As we come down here, we've got less hormones. That's going to drive down milk secretion per cell. And then we're just going to have the natural atrophy of the mammary gland. We're going to lose cell numbers as we work our way through. So looking at our beef breeds, we're looking at something like a 25-week lactation before they dry off. We try to stretch that out a little bit more than that. You're looking at 50 weeks, 48 weeks for our 305 lactation. Any questions on hormones? Let's start on immunity. I'll give you some highlights on that. With the time we have left, we'll get as far as we can go. So disease, whether an animal gets a disease or not, is the balance of challenge of that disease and the defense by the animal against that disease. So whichever is higher is whichever who wins. So the challenge to the animal is higher. We have to have a much higher defense to protect that animal than if we had a low challenge, we could get away with some things there. So who wins? Challenge is a function of the quantity of a disease-causing agent. The more that are present, the more likely we are to have a disease take hold, take over, and get clinical symptoms. The more we have, the more likely that is to happen. We also have to think about the quality of that disease-causing organism or the ability to cause a particular disease. So if we think about COVID-19, that became more and more sticky, had a greater ability to attack and be successful as it mutated. So the alpha strains were not nearly as effective at causing disease as the delta strains were. We think about something like measles. Measles has been in the headlines lately. If you're in the room with somebody with measles and you're not vaccinated, you will get measles. It's almost 100% transmission rate. It's a very sticky disease and moves very quickly from one person to another. So that's the challenge. What about the defense? Defenses take time and energy. Look at our defense budget for our national security. We can see that there's a lot of time and energy devoted to protecting our borders, to taking care of things. When we're thinking about the defense systems for our animals, we'll divide them into two categories. One is the passive defenses. We're usually considering things like barriers. So think about a castle. How is a castle defended? We start out with the moat, right? Going around the outside. That's a passive defense. Anybody here seen the Game of Thrones? Big battle at the end. What did they filled the moat with fire. Fire stays there as long as they don't go near it. There's nothing that can happen. So they're passive. The walls of the castle are barriers, but they are passive. They do not attack. Whereas the people up on top of the walls with the bows and arrows and those are active defenses, or catapults are active defenses. We're throwing something, we're projecting something, we're attacking in some way. So that's the other category, the active. We're thinking about the weapons, we're thinking about direct attacks, some sort of violent behavior. And we have the non-specific and the specific active. So 3 layers of defense, the passive, then the active, non-specific, then the active. So the first line of defense is our skin. It's a barrier that separates the outside from the inside. So as long as that remains intact, we will probably not get an infection. We have the nature, the structure of the skin to keep things out. We also have bacteria that live on the skin that are going to be in competition with the bad things trying to get through. So unless we have a cut, unless we have our skin drying out and cracking, that skin performs a very complete barrier. And we don't want to breach that if we don't have to. We have the mucous membranes. So think about your nose and your mouth. They're trying to flow mucus out of the body. So as bacteria come in, as debris comes in, that mucus should trap the particles, hopefully trap the disease-causing organisms. And then we have cilia on the top of the cells that are going to keep that, hopefully gravity, but also the cilia are going to keep that mucus flowing in the right direction. We think about the lungs, the ciliated cells of the lining of the tubes in the lungs are going to work our disease-causing stuff up, our debris up, and fight gravity and hopefully move things out so they don't cause us problems. We also have barriers that are related to chemicals, secretions of the body. So one of the barriers we have that a cow doesn't necessarily have, at least not initially, is the hydrochloric acid in our stomach. If we have a good secretion of hydrochloric acid, that should kill most, if not all, of the bacteria that come in. And then we digest them. We use them for an amino acid branch, especially if we're eating something like yogurt. Not all of them get killed, and that's how the probiotics populate the lower tract. So if you kill 99.99999, you still probably got 100 left that have the potential to colonize your lower intestine. Tears produce an enzyme called lysosome. It has antibacterial qualities. Has anybody heard of this before? Fleming, who isolated penicillin from mold, is the person who found this first. So there's a lot of, if you watch shows on that, there was a lot of jokes, political cartoons about him spanking children to collect their tears to provide injections to other people or antiseptic for surfaces and things like that. So there's probably others that we don't know about in these secretions. But again, they're just kind of flowing out. We have the active part where something is attacking in some way, shape, or form, or creating something as that particular disease-causing agent comes in. So we've got our white blood cells. We talked about the phagocytes that will ingest organisms. So they're going to attack, much like the army, charge, deal with anything they run into. They will ingest it, they'll digest it and spit it back out. We have antibacterial proteins that are going to either attack pathogens, disease-causing organisms directly, or will, so that's bactericidal. And then you've got bacterial static proteins that are going to stop those bacteria from reproducing. We have a compound like that in the teat end, in the wax that is secreted there, that is bacteriostatic. So that's why we usually only want to use the short nubs to do intermammary infusions, not the long rods that I grew up with, and they became my toys, my squirt guns running around the farm. So running around my pants with, 10 syringes in my pocket, squirting water everywhere. So if we use the short nub, we don't blow out that plug. We maintain the bacterial static properties of that wax in the T-den. We also have the inflammatory response that is going to help attract defenses to a particular area and help seal off those invaders from getting in. So the inflammatory response, we have a sliver coming through the skin, penetrating that outer barrier. We are now to the second line of defense. We have two different types of bacteria here, the red rods and the, looks like blue spheres. As they come in, we get next to a capillary. We got a number of, with the broken skin, we get a number of compounds being secreted. Histamines are going to start to be secreted. They are going to open up the capillaries. So anybody here allergies take antihistamines? So we're trying to stop that dilation in the nose that makes you sneeze. So capillary dilation, excessive amounts of that in your nose will swell out the tissues, make them itch, make you sneeze. We've got also prostaglandins are secreted, so are going to promote blood flow to that area. So we're going to open up the capillaries and then make sure those capillaries are filled with blood. That's going to attract white blood cells to that area, and they will start that first line of defense, the active, second line of defense, phagocytes attacking whatever they can. So we have the bacteria being consumed, digested. So they're going to be attracted to that area by the chemicals released in that injured skin. They're going to go there and start to destroy those invaders as best they can. So you're going to get a collection of dead red blood cells, pus. And if we do this right with the inflammatory response, the white blood cell response, we should have an abscess that is walled up and can drain out, hopefully outside the body and not inside the body. So you never want to puncture or have an infection lanced too early. If it's not walled off, if it's not formed an abscess, it can do more damage by lancing it than trying to drain it. So you need it to wall off, form a consistent abscess, and allow that. So the third line of defense is a specific immune response. So we have active immunity that fights against specific invaders. This is the basis for vaccinations. So thinking about that, the vocabulary we need to think about, we have an antigen, something that will activate the immune system. And this can be many different things on a disease-causing organism. So you might have the outer membrane pieces. You might have internal proteins that become antigens. The various proteins on the plasma membrane all can become antigens that these specific bacteria or specific white blood cells can learn to recognize and attack specifically. Measles has certain proteins that can be recognized and attacked. Those are antigens. What attacks those antigens are antibodies. So we have very, very large molecules that are produced to attack or recognize a certain antigen. Once that antigen is identified and attacked, it starts a cascade of response that hopefully will destroy the entire disease-causing pathogen. Bacteria, virus. So what do we need for a specific immune response? We have the initial responses from the first and the second line of defense. But what we're trying to do here with the specific is we're trying to learn to recognize specific disease causing organisms by identifying their parts. We could think about it. Has everybody heard the story about blind people going up into an elephant and trying to decide what it is? And then depending on what they touch first, they describe the elephant differently. So if they go up to the leg, well, it's a big trunky like thing. They go to the tail, it's big little bushy thing hanging down. If they go up to the side of it, it's a big wall thing, right? Or they get the trunk, it's a big squishy kind of rope like thing. We think about a bacteria, each of those spots could be a different antigen that could be recognized. So we need to be able to find for each disease causing organism a number of antigens that we can identify and specifically attack or assassinate that disease causing organism. Once we've learned what the antigens are, we have to have memory. We have to have white blood cells that are stored for later use. And this is what we try to do with a vaccine is we try to present things that the animal can respond to and then put those recognitions into memory that the second response to seeing that antigen, to seeing that part of a disease causing organism will be faster, more amplified than the first response. So the second draft of anything we do is slow, right? When we do something a second time, we usually can do it, at least for me, about half the time. Once we figured everything out, how to do things and get a system together, we can do a more effective job in a much shorter period of time that second time around. So we're looking at lymphocytes. These are the ones that are going to learn to do antigen recognition. They may or may not produce antibodies. They may or may not basically attack that antigen. So cytotoxic attack on infected cells. They will both be involved in the immunologic memory. And then we have a certain amount of regulation of that response. We don't want to overdo it. So we take B cells and T cells, and we're going to think about them as big categories. There are a lot of subtypes. We don't need to know them for this class. That's a grad level, or if you're going to be a doctor or something like that, you have to know the differences. But we've got B cells and T cells, and that's what we'll worry about. They are going to learn to recognize disease-causing organisms, react in different ways, and hopefully prevent that disease from happening. So if we start with the B cells, we've got memory cells, we've got B cells, we've got B plasma cells, lots of different types. They're going to learn what's bad. They're going to go and approach a disease-causing organism and learn different antigens on that disease-causing organism. When we've developed the vaccines for COVID, everybody's seen the cartoon of a COVID virus. What does it look like? Big pokey things. They got lots of Shrek ears sticking off them, right? That is what coronavirus, those spikes are meant to, because they have crowns, points on the crown. So the vaccines were designed to recognize those spikes. If anytime there's something with those spikes in your body, it really doesn't matter what it is, it's going to be a problem. Learn to attack it. They also thought that would probably not mutate as much as some of the other proteins in the thing there. So the job of the B cells is to build specific antibodies for our antigens that are presented. And then they store the blueprint of that antibody in the library. So it's going to take a little bit for them to figure out what that antibody is going to look like, how it's going to be shaped. But once they design it, they become memory cells and they sit in the library there waiting for that disease-causing pathogen to come back again. They can be there for years and years. If we think about the MMR, measles, mumps, rubella, most of it had that when we were 3 or 4 years old, and I haven't had to have a booster since. I probably will in the next few years. They said the immune system, as we start to fail, as we get older, maybe there's an opportunity to revaccinate, but got a couple years on that. So the B plasma cells produce antibodies. Antibodies is a synonym for immunoglobulin. And that is a term for a very, very large protein and carbohydrate molecule that is sticky to a specific antigen, sticky to a specific part of a disease-causing organism. So what is an antibody anyway? Why don't we stop now? It's what? 12 of. We'll start again at 7 of. So what is an antibody anyway? So we'll show you some pictures of that and figure things out.
Any, do you by chance know how large of an area our stage has for the sale?
20 by, is it 12 or 16?
Well, we'll go off the larger.
I think it's 2 sheets of plywood tall and 5 sheets of plywood across, so 20 by 16. Yeah, because we have 6 sheets, right? We carry the 4 in, and then the last 2 got to go in sequentially. So fit things in. Okay, so what is an antibody? So this is a schematic of that. We've got the sheets and coils of protein. So you've got a characteristic Y shape to them. The different colors represent different chains that are put together. So we're looking at that quaternary structure. So these are big, huge molecules, probably as big as you're going to find in the bloodstream. Probably myofibrils are bigger in the muscle cells, but these are about as big there. So we're looking at the structure. You've got this Y pattern. You've got the heavy chains on the outside. Then you've got the light chains on the inside of the Y. So if you go back and look at this, you're going to see the green and the yellow are going to be the light chains, and then you've got the red and the blue on that one are going to be the heavy chains. So big areas connected by a sequence there. What we care about is this part here. This part is modified by every particular or each type of B cell. And this variable region here is designed to be sticky to whatever antigen is present. So that's kind of a cool thing, that you're almost modifying the DNA to make sure that this particular antibody will see an antigen and then stick to it. So that variable region, we've got a constant stem here, and then basically 2 binding sites up top. Another look at that. We got the variable regions on the end where they are antigen binding sites. So whatever antigen we're dealing with, in this case, we're calling it a square. Anytime we see that square antigen, we can attack and attach. And we have a certain amount of flexibility in this area where that Y can open and close. So if we go through and look at each dot being a single amino acid, this is what our immunoglobulins might be mapped as. So you got your common stem there, the variable regions out there on the end, the heavy chain and the light chain. That's going to be designed to somehow balance the amino acids with hydrophobic, hydrophilic, different R groups to make them sticky to whatever antigen they're trying to learn. So we're looking at 5 different classes of antibodies. Most of you dealing with calves have heard of IgGs. These are the main ones in circulation. So B cells are going to create those, secrete them, and then they're going to be in the circulation of mom. And then when it comes time to make colostrum, they'll be taken out of the blood, transferred to the colostrum, and then we have an opportunity there, a small window for that gut to absorb those things whole. We need to be absorbed whole. If we start to digest them you're just going to have a source of amino acids, which is not the worst thing in the world, but that's not what they're meant to do. They don't transfer if they get digested or broken apart. So IgAs, these are going to be in the mucus that is secreted from like your nose and your mouth. So produced and secreted. IgEs associated with the allergic responses and parasitic infections. IgMs, these are involved in early immune responses, getting things started. And then IgDs, they're found in B lymphocytes and needed for the maturation, the effectiveness of those B cells. So what do those antibodies do? They're going to see an antigen that they're trained to learn to recognize, and they're going to go over and they're going to stick to them. So in this case, you're seeing the antigen here. We got the triangle-shaped antigen. We got the triangle-shaped sticky spot on the variable end of our antibodies. And we attack that. So this can immobilize, identify. A lot of times those antibodies going over and sticking to that disease-causing organism, we couldn't see it before, but now because of the body's antibodies on there, we can now see it, and the rest of the immune system can attack it. So it's almost like tagging our antigen, whatever it is, or disease-causing organism. So if we're trying to get a good response, we'd like to have multiple antigens for one disease-causing organism. So we have, in this case, 3 different types of antibodies that will react to different antigens. And then we're going to stick to them. What we're hoping, I think, is we start to get the disease-causing organisms kind of caught up in a net, kind of stuck together, kind of immobilized, so it's easier for the immune system to identify them, attack them, and get rid of them. So after they're stuck, then what? Spleen's going to filter things out. The rest of the immune system will come over and take care of them, kind of like Spider-Man hanging the criminals, right? Everybody who's seen the Spider-Man movie, right? We hang the criminals, and then the cops come in and take care of them from there. So the rest of the immune system will come in and take care of the criminals as they need to. T cells, they have a little bit more personal approach. They're still going to learn to recognize antigens. They're going to learn what's bad. And then they are going to design their attack for that particular organism. So this is more of a personal approach, a personal attack. The T cells are going to attack directly. So they're going to wait in the library until they're called, wait till we see that antigen, and they're going to attack directly. So again, they're going to have memory that lasts years and years. So we got B cells that kind of stand at a distance and throw antibodies. And then we've got the T cells that are going to attack that antigen directly. So when we think about our T cells, we have cell-mediated immunity. We have a number of T cells working together. We've got the cytotoxic cells are going to recognize that antigen. They're going to find, grab, or destroy that antigen and whatever is attached to it. You've got helper cells that are going to go, hey, over here. You need to come over here. The cytotoxic, come on over here. They help tell the cytotoxics what to do because they're not the smartest ones in the world. And then we have the suppressor cells that are going to, okay, break up the fight. That's enough. We've done enough damage. We don't want these cytotoxic cells going crazy and start attacking things that they shouldn't, the body, the calf's own cells. So they're going to limit the cytotoxic response. So we're thinking about T cells, it's a direct hand-to-hand fight. So if we're going to compare the two, if we go to the Marvel comic universe, I would think Spider-Man versus the Hulk. So Spider-Man, or whatever version of Spider is, the male, female, if you've watched Into the Spider-Verse or anything like that, you've got webs that are going to equal the antibodies. They're thrown out and hopefully immobilized and help destroy the culprit, whereas Hulk does what? Hulk smash. So it's a more personal approach. So moving through, so B cells versus T cells. So this is a more formal presentation of that. So you've got the B cells kind of here. So you're going to have B lymphocytes secreting antibodies. They're going to attach to the antigens on the outside or inside of that invader, and they're going to hopefully block infections and help eliminate those extracellular microbes. With our T cells over here, you can see that the T cell here, the helper cell, is kind of holding that cell down. And then we've got the cytotoxics that are going to come in and basically vomit all kinds of digestive enzymes on the invader and destroy it. So taking the infected cells, making sure they get destroyed, that more personal Hulk smash approach. So it's a coordinated attack. You've got a gang of T cells that are going to eliminate that invader. So challenge versus defense. We got a high challenge. We got a tough environment. We have to establish a good defense. So if we're thinking about the development of a newborn calf, what's going to happen? As I found out this semester, it's the same thing with our equine. They get colostrum and all that stuff as well. Need more chalk. Okay, so if we take our newborn calf and we think about time down here and level of immunity over here. On the y-axis. So we know that a calf is not going to really develop its own immune system till, like, say, 2 or 3 months. And then they're going to work on that first line of defense and learn to produce their own B cells, their own T cells. So the problem becomes, and I'm oversimplifying here, is getting from birth to 3 or 4 months. So how do we get there? So we can think about colostrum management, making sure our calf gets a good quantity of high quality colostrum, and that's going to give her a whole bunch of antibodies that mom made. So they usually call it passive immunity, but really it was made by mom's third line of defense, the active specific. So you got to be careful with how you use passive immunity. Those IgGs are not part of the passive immune system of mom. So if you go there, we hope that we start out with a high level immunity and we have something where those IgGs last a long time. If our environment is such that the challenge is low, that's likely to happen. If the challenge is high, what's going to happen? You're going to use up those antibodies very, very quickly. And you're going to have no way to replace them in the short run. And then if you get rid of all that immunity, you're going to be vulnerable in this area right here, high vulnerability. The same thing happens with if we don't get enough colostrum into that animal, but we use it up at the same rate we did before, you're still going to have that area in here where that animal is going to be vulnerable. So if we look at our calves' immune system, these are big molecules. They need to be transferred without digestion, and that only happens for a very short period in our calves. So this is a classic study done in, I think, the 1970s, where we take the percentage of ingested immunoglobulins that are absorbed versus the hour from birth to first feeding. So if we feed that animal more or less immediately, we're going to get 100% absorption of those IgGs. If we wait for 24 hours, we're only going to get 10% absorption. So we have a gut that is open in our calves, and we have holes or pores in that gut that allow the IgGs to move through. The challenge with those is whatever goes through first gets absorbed first. So we want colostrum to be the first thing that goes into the mouth of a calf so that we can absorb those proteins whole. Those holes are big enough for viruses and small bacteria to go through as well. So if that calf is born and the first thing it does is swallow a whole bunch of manure, then that's going to be the first thing that's absorbed. And that is going to make that animal sickly and not there. Once something gets absorbed through that hole in the gut, it closes behind. So if manure is the first thing in there, you're going to have a whole lot of gut closure and you're going to have no transfer of IgGs. The gut will close itself off automatically over the first 2 days. So our thoughts about colostrum are what? We need to get as much as we can, as fast as we can, the best quality we can. So best we can get, best colostrum we can get, as much as we can get into that animal. So what are we doing up the dairy? I think it's a gallon, right? Or is it 3 quarts? That first feeding, we're trying to get as much as into that animal as we can. We're trying to do it as fast as we can. The standard recommendation is before 6 hours. So you saw if we're at 6 hours, we're still doing pretty good. So if I can get it in this window, that's where I need to be. And this is the study that does that recommendation. Everybody's seen those joke photos where the calf is coming out and somebody's sticking a bottle in them. So as they're halfway out. That's an extension joke. So as much as we can, as fast as we can, we're trying to get the IgG levels up in that calf, in the blood. That's an indirect measure of how much IgG was transferred. So the higher we can make that level, the better we are. So if we're looking at the system here, we want to get that colostrum as high as we can, as much as we can, as fast as we can, get as much transfer as we can, and then we want to maintain the defenses, keep the challenges low till she can develop her own immune system. Particularly if we screw up in here, what is this going to make difficult? Weaning. That's a stressful time. That's going to suppress whatever immune system we have. And it's going to make that animal very vulnerable at weaning to problems. So we're looking at vaccinations and how do they work. So their job is to develop B cells and T cells. What do we need for that? We need a working immune system, a working third line of defense. So that doesn't occur till here, which is why we really don't do a lot of vaccinations till they're 4 to 6 months old, when they have a working immune system that's going to respond effectively. I'm not sure what this blue stuff they call it a vaccine in the nose. I don't know is it's generating B cells and T cells. It might just be coating things. It might be promoting the secretions of the IgMs and things like that. But it's not a vaccination that we'll talk about here in the truest sense. At least I don't think it is. So how do vaccinations work? I did that already. So vaccination. What I need to do is introduce, if I vaccinate out here, I'm introducing specific antigens. I want to develop B cells and T cells, that third line of defense. And then hopefully with the vaccination, I will increase the immunity. So vaccine is going to give us one more level of immunity. Moving up. So we're going to draw that up. So what do we need for that to happen? We need to have an animal that's not stressed. So we don't want to move animals and vaccinate them at the same time if we can avoid it. Unless it's a very low stress kind of thing. I know growing up, we used to move animals. We put all animals in a trailer. And then when I was working for my dad, I'd be surfing the top of the animals, giving shots and marking them as I went. So that was probably not ideal conditions for giving vaccines from a efficacy standpoint. So we need animals that have a good plane of nutrition, not stressed, follow the vaccine directions so we get a good vaccine response. What's the advantage of the vaccine? Two main different types. We have killed, and then we have modified live. So we have a certain volume of injecta. We've got dead disease-causing cells or parts therein. We inject those antigens are presented to the immune system, and then we develop a moderate response in B cells and T cells to attack that should it ever show up again. So the idea behind a vaccine is we're going to create antibodies recognition before we have to deal with the full-fledged attacker, the full-fledged bacteria or virus that causes the disease. These usually require a booster. So we do it once and then we go through and we give it again, and then we get a higher response each time we do it. So think about probably tetanus vaccine. We get boosters for those on a regular basis. Although I have a friend that works as an ER doc, and she says nobody that's ever got the initial tetanus shot has ever gotten tetanus in this country. We do the booster every 10 years. I think anymore, it's about another way to charge for your services. So modified live, you have an organism that's been crippled or compromised in some way. That gives a stronger immune response because it's alive and can sort of replicate. So it's going to challenge the immune system more. So you inject what it is, it's going to multiply, but not cause the disease per se. And you get a stronger immune response. So this is all about activating that third line of defense. So if we're thinking about it, I got a calf that's got no colostrum. How am I going to raise that animal? Sterile facility, at least in theory, right? You can't develop a rumen in a sterile facility, although they've done it. But you're going to isolate that animal. You're probably going to feed it first. You're probably try to keep it away from all the adults, things like that. You're going to give that animal a high plane of nutrition to maintain the first and second lines of defense. So maintain their skin, maintain the white blood cells that are non-specific. You're going to put that in an environment, and we got the extreme example though, sterile environment has no challenge in it. You're going to try to keep it into as clean an environment as you can. So, and probably not contact with other calves. So take advantage of what you have with the first and second line of defenses, and then hopefully you can get over that hump until they get to 4 months old and start taking care of things on their own. So mastitis, somatic cell, we have the inflammatory response, draws white blood cells to the area. Somatic cells are white blood cells of the body. So they are an indirect indicator of infection and not a perfect indicator of infection. Sometimes with high somatic cells, you cannot get a culture that shows a bacteria causing the problem. So there's a bunch of stuff. I think I posted this handout. If you're interested in viruses, COVID-19, we went through, I think I started the semester that following spring with all the things related. This one's a good one. Talking about how diseases spread among a group of people, like hands and noses and sneezing and all that stuff. With the sneeze, or a cough, how much actually gets spread out. So the art of sneeze and flu friction, these spread severely. So any questions? I'll leave you with one last thought. Why do we find so many teeth in the first calf? Pen 1 up there. Why do we find so many teeth in the water tub? That's when they lose their teeth. So we have deciduous teeth. Don't look a gift horse in the mouth. Same thing could be said of a cow. You can age a cow by the number of baby teeth or deciduous teeth it has, and then older teeth. So we look at 2 years, we're starting to replace things, 2.5 years, and then we see the wear on those teeth as time goes by. You can start to figure out how old that animal is, the eruption and the wear. So by the time they get down here, they've lost all their paddles. Things continue to grow, hopefully at the rate they wear, and everything gets matched up. So you've got deciduous teeth. You're starting out with a few in the front, and then we're going to add more teeth as the animal gets older. As those teeth, if you want to take a look at a few, I've had the dairy collect teeth as they find them. Those teeth wear, continue to grow up through, and then they fall out as hopefully about the time they're worn out. So you can see, depending on where you are, when the teeth first come out, you've got that tip there. As they wear down, we get different patterns. You've got the double pattern here, the groove in the middle. Eventually we get down to the point where we're got a more of a solid approach. So I think those things are up there. If you want to take a look, they're from the Atlas of Bovine Anatomy. We didn't get quite enough time to go through that. So got teeth up here if you want to play with them. I hope you learned something this semester. We'll see you on Monday, I guess. So.